everything I'm going to say now are the things you are going to see as far as this 2024 NECO exam biology practical is concerned. So all I want you to do now is to get your pen and paper ready. And please, if you have anybody that will be writing NECO this period, all I want you to do is just share this video to those people so that they can partake in this lecture. Yeah, good day viewers, welcome to my channel. If today is the first time of visiting my channel, I want you to do well by hitting the subscription button and also turning the subscription bell on so that anytime we post in this channel, you can be able to get notification to enable you watch our videos anytime we post. So on today's edition, I'll be giving you some of the specimen as given by NECO. Yes, 2024 NECO biology practical. That is what we shall be looking at today. So all I want you to do is just to pay attention in this teaching. If you have any question you want to ask, I want you to drop your comment on the comment section below. And also I want you to understand that everything I am going to say here are the things you are going to see as far as this NECO exam biology practical is concerned. Yes, everything we are going to say here, you are going to see it. It is a must. It is a must. So all I want you to do is to get your pen and paper to write down something. So let us take a look at the specimen one after the other. So from here, I have specimen from Specimen A down to Specimen H. From Specimen A down to Specimen H. So I am going to be giving you the questions that you will see as far as the specimen is concerned. Okay, so here we have Specimen A, which is onion bulb. Onion bulb. Now there are things you'll be expected by the NECO exam. So these are the things you'll be expected to know. One, you should be able to know how to draw onion bulb. You should be able to know how to draw onion bulb and label it fully, because from there you can see the onion bulb on the screen where we have the structure of the onion bulb. We have the bulb apex, we have the tunic leaf, then we have the bud, we have the basal plates, and then the roots. These are the labelings that you are expected to put down as far as the drawing is concerned. So go and learn how to draw. Okay, it is very, very important. Now another thing you should note there is the propagation. How is onion propagated? How is onion bulb propagated? Now onion bulb is propagated vegetatively. Yes, onion is propagated vegetatively. That is one thing you should understand as far as the onion bulb is concerned. So we will not be dwelling more on this onion bulb because there are few questions that you will be expecting as far as the onion bulb is concerned, which I have mentioned. Now the next one there is Specimen B. Specimen B is ginger. Yes, the ginger. So you also have to learn how to draw the ginger. Now here we have the area shoot, we have the scale leaf, we have the bud, we have the node, we have the internode, we have the adventitious root. Now also note the botanical name of ginger. The botanical name of ginger. Here we see that the botanical name of ginger is Zingiber officinale. Zingiber officinale. Now ginger is propagated vegetatively using its rhizome. Ginger is propagated vegetatively using its rhizome. So this is ginger. This is ginger rhizome. So this is what we use in propagating it. Okay? This is what we use in propagating a ginger. And it is propagated vegetatively. Take note. So learn how to draw a ginger rhizome. It is very, very important. Now let's take a look at Specimen C. Let's take a look at Specimen C. Now here we have the structure of Specimen C, which has the apical bud, you have the periderm, we have the lateral bud, which is also known as the eye. We have the node, we have the scale leaf, we have the lenticel, and we have the stem end. The stem end. So that is the structure of specimen C. Now, the next one there is propagation. The next one there is propagation. This specimen C is propagated by seed tuber. It is propagated by seed tuber. Now let's take a look at diseases of specimen C. Diseases of specimen C. One of the diseases of specimen C is fusarium. Fusarium and Fusarium sambucinum. Fusarium sambucinum. Now we also have the scab. We have the cutworm, we have the Alternaria solani. Alternaria solani. So all these that I've mentioned are diseases that are capable of attacking or affecting specimen C. Okay, now the next one there is a potted bean seedling of week old. A potted bean seedling of a week old. So as a teacher, all you need to do is a week to the biology practical. All you need to do is get beans, plant it in a pot or in a petri dish. You know how to do it. Plant it there. Now that very day, the biology practical will be written. You bring it out and place it. So on the screen, you can see the structure of a week-old seedling of beans. So now when you take a look at the structure there, you will identify the leaf. It has the leaf. The leaf is already out. Now it has the cotyledon, which is attached to the stem of the leaf of the plant. Which is attached to the stem of the plant. Now we have the stem and we have the roots. Now another question that you will be expecting as far as this NECO is concerned is the type of germination. Yes, they will ask you the type of germination of this bean seedling, which is in a pot. Now the type of germination of this seed leaf is called apical germination. It's called apical germination. So why is it called apical germination? The reason why it is called apical germination is because the cotyledon is raised above the soil. It is raised above the soil. It is not that kind of cotyledon that will remain down inside the soil. Any plant that carries the cotyledon, which is the seed which the plant germinated with, up above the soil level or above the root, such is called apical germination. That is why we call it apical germination. Okay, now another question, very important question that they will never fail to ask you as far as NECO exam is concerned is the type of cotyledon. Is the type of cotyledon. What kind of cotyledon does bean seedling exhibit? Now, bean seedling exhibits dicotyledon. It is a dicotyledonous plant. Yes, bean is a dicotyledonous plant. Reason why it is a dicotyledon plant is because it is made up of two seed leaves. It is made up of two seed leaves. That is to say that when it begins to germinate, it does not just germinate like this. It germinates like this. Then that is how it germinates. So the leaves are usually two. It comes out in two, just like this. Just like this. Now, after some days, it will now split like this. Another one will shoot out and it will germinate as it will spread like this. That is how it continues until it completes its life cycle. So that is why we say that bean seedling, that is the germinating or the seedling in the pot, which is specimen D, exhibits dicotyledon. It is a dicotyledon plant. Now this plant is propagated by seed, meaning that we plant it by seed. We use seed in planting it. propagation, another word for propagation or simplest term for propagation is planting. So we plant it using the seed. Okay. Now let us look at specimen E. Let us look at specimen E. Now specimen E says a week old of Guinea corn. A week old of Guinea corn. So you look at the structure of the Guinea corn. That is the diagram on my screen. So from the diagram on my screen, I have the shoot, the leaf blade, the collar, and the roots. The collar and the roots. That is the diagram of a Guinea corn. So now let's take a look at the cotyledon. Let's take a look at the cotyledon. Now here, Guinea corn exhibits monocotyledon, which is the specimen E exhibits monocotyledon. Why do we say that it exhibits monocotyledon? Reason why we say it exhibits monocotyledon is because from my first explanation, I told you that these plants, they have dicotyledon have 2 seed leaves, but monocotyledon only have 1 seed leaf. Okay, it has 1 seed leaf. So how it germinates is 1 seed leaf will grow out, then after some days it will spread like this. Another one will come up, it will spread like this. Another one will come up, it will spread like this. One will come up, it will spread like this. Why that of dicotyledon comes in this form? When it germinates, it comes in this form. After some days, it spread like this. Another 2 comes up, and after some days, it spread like this. That is how it continues. So that of Guinea corn, from our experiment, we identified that it is a monocotyledon because it only have 1 seed leaf. So teachers watching me from this channel, I want you to take note of that. Make sure you carry out the practical if you are not still convinced. Okay, now let's take a look at the type of germination. What kind of germination do Guinea corn exhibit? What kind of germination do they exhibit? Now, from the experiment we have conducted so far, or from the one drawn on the screen, you will discover that the seed lies below the soil surface. It lies below the roots. Even sometimes you don't even see the seed. Okay, so that is the reason why we say that specimen E exhibits hypogal germination. It exhibits hypogal germination. That is to say that we have 2 types of germination, which is epigal germination and the hypogal germination. Epigal, like as I said earlier, epigal raises the cotyledon above the roots, while that of hypogal sustains the seed below the roots. So that is by the way. Now I want us to look at a very, very important question that will be coming up. Now this question is factors that can affect the growth or germination of specimen C, specimen D, specimen A, specimen B, and lastly specimen E, which is specimen A, specimen B, specimen C, specimen D, and specimen E. Factors that can affect their growth or their germination. Now, one of the factors that can affect their growth is temperature. Temperature is very, very important as far as germination is concerned. So when there is a very low temperature, the plant will find it difficult to germinate. So that is one of the factors that affects the germination of these specimen that I've mentioned. Now, another factor that affects them is low relative humidity. Low relative humidity is also a factor that can affect their germination. When the amount of water in air is very, very low, or when there is no water in air, it can also affect their germination or their growth. So I want you to take note of that. Now, another one there is soil type. The soil type. Soil type is a very important factor that affects the germination or the growth of specimen A, Specimen B, Specimen C, Specimen D, and Specimen E. Yes, soil type. And you all know that we have 3 types of soil, which is the loamy soil, clay soil, and the sandy soil. So each of the soil that I've mentioned has a special effect it renders on each of these plants that I've mentioned. So soil type is a factor that can affect their growth. Okay, now is sunlight. Sunlight. Of course, you know that all of these plants that we have mentioned are all green plants. They possess green leaf. Yes, they have green leaf. So for the fact that they have green leaf, which means they need sunlight in order to manufacture their food. In order to photosynthesize, they need sunlight. So when there is low sunlight or when there is no sunlight, you can see that their growth will be what? Affected. It will be affected. So that is it. Now another one there says viability. Viability of the seed, tuber, or the leaf. Viability of the seed or the tuber or even the vegetation is very, very important. What I mean by viability is how healthy it is, how healthy the seed or the tuber or the tubes is. Now, if you happen to plant any of these specimen A, B, C, D, or E, when the seeds are not healthy, when the seeds are not viable enough, you will see that the plant will not germinate. That is another factor that affects it. Now, lastly, from here, disease. Disease is also a factor that affects the germination or the growth of these plants that we have mentioned here. So I want you to take note. Please remember, do not skip this video, because anytime you skip my video, you will miss one step or the other. And remember, if you have any question you need to ask, don't hesitate to do so, because it is very, very important, and I promise you that I will give you answer to every question that you ask on the comment section. Okay, now let's move over to specimen F. Let's move over to specimen F. Now here we say specimen F here says head of a pigeon with the neck. Head of a pigeon with the neck. Now if you look at my pigeon, this is a preserved pigeon. We have already preserved it. I believe this pigeon has lasted up to 7 years. It has lasted up to 7 years. So, but here now, they are demanded we should get the head of a pigeon with the neck intact. So for now, there is no way I can cut the neck, but just take a look at the structure of the head and the structure of the neck. Now, the question is, draw and label the structure of specimen F, which is the head of a pigeon. Now, after drawing, you will have to label it. Now, from here, we have the beak, we have the eyes, we have the nostrils intact, and we have the neck. Do not forget to label the neck. Okay? Do not forget to label the neck. Now, let's take a look at the adaptation or the functions of all these that we have mentioned. The functions of these parts that we have mentioned. Now, the eyes is used for sight, vision, or seeing. Whichever way you want to write it, it is correct. It's either you write it as vision, sight, or foreseeing. That is the function of the eyes. Now we have the nostrils. The nostrils is used for gaseous exchange, that or used for breathing or used for respiration. So anyone you want to write, it is correct. Now we have the beak. The beak is used in feeding. They use it for feeding. It's adapted for feeding. It's adapted for feeding. Now, another question that they would not fail to ask you is which of the food is the specimen H, that is the mouth, adapted for? Which of the food is this pig adapted for? Now, if you look at the structure of the pig, it is pointed. It is not curved like this, like that of the ego. Like that of the ego. No, it is pointed. That is to say that the food that is adapted, that this mouth is adapted to, is grains. Yes, it is adapted to grains. Example of grain, it is adapted to, let's say, maize, corn, beans, rice. So all these are food that this specimen H is adapted to. So as you can see the head, you can see the beak. So that is by the way. So now let's take a look at another specimen. Please let me hang this so it doesn't fall off. Let me hang it. Okay, so having done that, having explained that, now let's take a look at the next specimen, which is specimen G. The specimen G here is Atlas. That is the name, Atlas. Please remember, the way they ask this question, they may ask you to identify, though I will still explain that further. When you are asked to identify simply means you should write the name. That is all they want from you. So now here we have the Atlas. Now, where is this Atlas located? That is the question that they will not fail to ask you. So take note on that. The Atlas is found in the cervix. That is the cervical vertebrae. And where is this cervical vertebrae located? It is located around the neck. It is found at the neck region. It makes up the cervical vertebrae. So now there are two bones that makes up the cervical vertebrae, which is the atlas and the axis. Those are the two bones that makes up the cervical vertebrae. Now, remember I told you that the cervical vertebrae or the atlas is found around the neck region. That is how you write it. It is found around the neck region. It is a component of the cervical vertebrate bone. Now let's take a look at the function of this atlas, which is the specimen G. What is the function of this atlas? Now we say that the function is it allows movement in one direction. It allows movement in one direction, which is the movement of the neck in one direction, which is nodding of the head. So the bone that enables you to nod like this is known as the atlas. Why the other bone, which I mentioned earlier, which is the axis, allows you to rotate your neck in sideways. So that is by the way. So take note, the function is allows you to nod your head. Allows the organism to nod the head. Okay. Now let's take a look at the specimen H, which is the lumbar vertebrae of a rabbit. Remember, both the specimen G is that of a rabbit, and this one now is also that of a rabbit. So now, where is this lumbar vertebrae located? That is your major focus. Where is it located? Now, the lumbar vertebrae is located at the upper abdominal region. It is located at the upper abdominal region of rabbits. That is where the lumbar vertebrae is located. And it is 7 in number. Some textbooks will tell you 6 to 7 in number. Or whichever way, just note that it is what? 7 in number. Take note. The question must come. Okay? The question must come. So, I believe I've been able to do justice to this specimen. Please remember, if you have any question you want to ask as far as this specimen that I've mentioned is concerned, please drop your comment or drop the question on the comment box, and I promise you that I will give you answer to every question that you ask. And remember, if you need any assistance, as far as this NECO exam is concerned, all you need to do is just drop just chat me up on my WhatsApp group, and I promise you I will add you to our group where we give you all the answers to the question a day before the exam. So just follow my instruction, and I promise you will never regret it. Thank you very much, and God bless you.